hematopoiesis, the hematopoietic system, how cells evolve in the hematopoietic system. And we're going to do that in the context of some data from mice, where you can parameterize a model in great detail and see what we can learn about how cells evolve and how this can give rise to cancer. And then in the end, we're going to move to spatial dynamics of stem cells and see how spatial restriction and spatially explicit model can change some of the results. But before I get going, I want to say that it's wonderful to be at a conference that celebrates the achievements of Siv. Siv has been a great supporter and friend over the years. I always look forward to seeing him in conferences, to hanging out. It's always a lot of fun. And we were just reminiscing that in one particular conference, we got to play basketball together on the team, which was a very fun experience. So if Siv ever gets tired of mathematical medicine, there's a career waiting for him in basketball. All right, so let's get going talking about the hematopoietic system. So this is a basic picture of how the hematopoietic system is organized. So that's blood, basically. So blood is maintained by the hematopoietic stem cell, which is in bone marrow, and the stem cell gives rise to various downstream cell compartments, progenitor cells, lymphoid and myeloid progenitor cells, and these then differentiate further to more differentiated cells, eventually leading to those that perform their function. So there's a very clearly defined differentiation hierarchy in the hematopoietic system, and it's been worked out pretty well experimentally. So we know what the cells are doing, and we know quite a lot that is very useful for model parameterization. This is just the same picture, kind of, and it shows you where things can go wrong in the system. Cancers develop in the hematopoietic system. You get a variety of blood cancers, and there are different kinds of blood cancers that all have different characteristics. Some of them arise kind of very early in the stem cell compartments, for example, CML or AML, or myeloproliferative neoplasms. And we'll be thinking about these kinds of cancers in this talk here. There are others that arise downstream, such as CLL. We're talking less about that. What we're interested in this talk is to look at how does evolution proceed in the stem cell compartment, in these sort of rather primitive cells. How can mutants evolve that predispose people to cancer, and what can we learn from models? And so a particularly interesting phenomenon in this context is what we call clonal hematopoiesis of indeterminate potential. And what this means is basically, as people age, their blood starts to show cells with certain mutations. And interestingly, these are mutations that can be characteristic of cancer. So you find those in a variety of blood cancers, but in this case, they do not, it's not cancerous. It's a healthy hematopoietic system, but these mutants tend to show up. And that's called clonal hematopoiesis. And so as people age, these mutants pop up, they're there. So what do? They, in a very small fraction of people, these go on to develop cancer. That's a relatively small risk. In a greater proportion, majority of these patients, they have cardiovascular complications or metabolic complications from the presence of these mutants, because these mutants can cause inflammation and they can cause chronic health issues. So even in the absence of cancer, these mutants are a problem that need to be dealt with medically, but they also provide a basis, obviously, for carcinogenesis to proceed. So we want to understand how do these CHIP mutants evolve? What are the selection pressures that account for the rise of the mutants? And if we understand these, can we treat these, can we have evolutionary treatments to reduce CHIP mutant burden? So there's lots of existing mathematical literature on this already in a variety of contexts. What we did, we looked at the evolution, particularly in stem cell models, applied to some very recent data from mice that allow very detailed quantification of the kinetics of the hematopoietic stem cells. So this is just a bit of more detail about the CHIP dynamics, the dynamics of these mutant clones. It's kind of unclear exactly what they do and what their fate is. So some people argue that they kind of grow exponentially, but even if you look at these data, they fit exponentials to that. It kind of doesn't look that convincing. Then other people group them into, they can be growing these mutants, they can be declining these mutants, or they can be static. So sometimes the mutant will come up, it can just hang around there, and at the end of the day, it disappears, it's gone. In other cases, it stays steady, and yet in other cases, it keeps growing and growing and growing. And so what does it all mean? We don't really understand all that much about it. So this is sort of a summary of what our understanding is. You get an initial rise, get some steady levels, and they rise further, they stay steady, or they decrease. We don't really want to address these complicated dynamics. What we want to address in the model is to understand, how do these mutants arise in the first place? How do they evolve? What does it take for a mutant, when it gets generated, to start expanding as a clone and to be initially successful? So that's the question that we asked. And so we did this. We tried to answer this question with mathematical models parameterized by data. So let's get into the mathematical models. Before we go into the hematopoietic system, let's talk about just stem cell models in general, and then build up the complexity. So what are stem cells? Can divide in a variety of ways. In some cases, people talk about asymmetric cell division, where one stem cell gives rise to one stem cell in the differentiated cell, and so it maintains the population while keeping the stem cells constant. But there's more and more evidence that this is a division pattern that applies in humans quite a bit, that stem cells can either give rise to two daughter stem cells or give rise to two daughter differentiated cells, and this happens with given probabilities. And if they balance each other out, then the stem cells are maintained in the long term, and the tissue is maintained through differentiation. So this is the kind of stochastic stem cell self-renewal and differentiation model that we're using. It's going to become more complicated as we apply to the hematopoietic system, but this is essentially sort of the core of our assumptions. And there are data that support this sort of model of stem cell dynamics. So this is on the most primitive level. This is a model like this. It's an ODE that describes these dynamics. Basically, whoops. Let me go back here. The stem cells, they divide with a given rate. Upon division, that division can be either self-renewal with a probability p, or differentiating with a probability 1 minus p, and the differentiated cells die with a rate d. And so in this particular system here, it's not a very stable system. Your p, the probability of self-renewal, has to be exactly 0.5 in order for the system to be stable, because if it's bigger than 0.5, then the population grows, because the self-renewal is bigger than differentiation. If the self-renewal probability is less than 0.5, the population declines, because cells differentiate out more than they self-renew, and so it needs to be exactly 0.5. So self-renewal is a very important parameter here. So the way to get around this, to get a more reasonable model for homeostasis, is to include feedback into these models. So for example, you can assume that some cell type, like the differentiated cells, they secrete some factors that will reduce the self-renewal rate. So if there are lots of differentiated cells out there, they stop the self-renewal, they increase the differentiation, and the population stops growing, and you go to a stable equilibrium. That gives you homeostasis. You can also have, it has been shown that you can get feedback on the rate of cell division, but this is not important for stability of the system. So in this model, what is important for stability, for maintaining a stable equilibrium, is to have some sort of a feedback on the self-renewal probability to shut down self-renewal as the population gets bigger. So that's the basic model that we deal with. So now we adapted this model to make it more complicated, because we're talking about the hematopoietic system, and we know quite a bit about the different stages. So there are the long-term stem cells that are sort of at the top of this hierarchy. They are the ones that are thought to really maintain the whole hematopoietic system. They are thought to divide pretty slowly. They self-renew. They give rise to downstream compartment, which are the short-term stem cells, short-term hematopoietic stem cells, HSC. They're still stem cells, but they divide a little faster, probably, and are less able to self-renew. And then it goes down the differentiation pathway. You get the progenitors. These are MPP, multipotent progenitors. They still have good self-renewal probability potential, and differentiate further. Then they start branching off into CLP and CMP. which is the common myeloid progenitor, common lymphoid progenitor, and then it continues onwards, but we don't have much data on these, so we consider these the final compartments in our model. So we basically tailor the model to have as much detail as we have data and no more. And so the model is given by differential equations, and these equations are essentially the same as I just showed you before with a simple model. It just includes these different compartments. Now it's a little bit more complicated. And again, we assume that there's feedback going on, negative feedback from cell populations onto the rate of self-renewal, like I had it in the simple model. And there can be various assumptions about that. They can, all the feedback factors could in principle be produced by, this is not working very well, in principle be produced by any of these cell populations and feedback on any of the self-renewal rates. And we've done a general analysis, but to show you some results, I'm going to use a model that is specific and that has a specific feedback loop, and we use the simplest feedback loop where each cell population feeds back on itself. So the long-term stem cells secrete factors that reduce their own self-renewal probability. The short-term stem cells secrete feedback factors that reduce their own self-renewal probability, and so on, the MPPs as well. And that gives you a stable equilibrium, and that's what we work with. And the feedback loops are given by these expressions here. So the self-renewal probability P0 of the long-term stem cell depends really on two things. There's a self-renewal probability in the absence of feedback, which we call C0, and then there's a strength of feedback, which we call H0. And so these two parameters then determine your actual self-renewal probability, which is a function of feedback.
H0 is 0, that means we don't have any control.
Sorry?
H0
is no control, yeah.
So what is the reason to take this formula? Is there biologically, because PI could be, in your expression, could depend on different parameters, right? PI depends on both these compartments.
Right.
What is the reason? Is there any biology?
What is the reason for what?
Yeah. What is this exact form of?
Oh, this is just an example. This is an example of a specific feedback loop we're modeling. You can have the feedback factors produced by different cell populations. They can be produced by the downstream cell populations, and they can feed back on the upstream. You can do it in various ways, and we analyze it in more generality. It's possible. Yeah, we've done it. In fact, I'm just for demonstration purposes, I'm using that particular model. Yeah. So this is how this works, and for each compartment, this is the same applies to each compartment basically. So now we wanted to parameterize the model, and we do that, which what is called neutral label propagation data from mice, and these have become available relatively recently from a group in Germany, and we then use the parameterized model to first of all examine some basic cellular dynamics, and then to go on and explore the consequences for mutant evolution. So these are labeled propagation data. What do you do? I mean, this was done by the group of Thomas Höfer in Heidelberg. You put a label that you can induce into the long-term stem cells over here. And then over time, you watch that label move from one compartment to the next. So it goes from here, it propagates to all the different compartments, because the long-term stem cells will divide and will differentiate out. The label will go with them. And so you can see how the label spreads through all of these compartments. And then you get something like this here, a graph of the label frequency of the cells over time. Now you can derive equations that describe the label frequency over time from the model. This is what we have done. I'm not going to go into great detail here, but you fit that to the data as the label propagates, and then you use the best fitting parameters as an indication of what biologically reasonable parameters could be. And the fitting wasn't that complicated. There didn't seem to be too many issues with parameter identifiability. So we got these estimates here, and they look reasonable. So you can see that the division rate, the estimated division rate of the long-term stem cell is the lowest. The division rate goes up. Then you could estimate the self-renewal probability of the different cell compartments at equilibrium. This is all at equilibrium, at homeostasis. You estimate the death rates, and the equilibrium number of long-term stem cells is kind of known in the paper that we were referring to, about 17,000. of those. So that gives you basic parameters to work with. You have to also parameterize the feedback, which is more complicated because we don't really have good enough data to do that. But we know that at equilibrium, certain values hold, like the self-renewal probability or the number of stem cells at equilibrium. And so we have these parameters, the self-renewal probability in the absence of feedback and the strength of the feedback. And you can find a combination of the two that matches the observed experimental values. And then we have some sort of possible feedback scenarios that are consistent with the experimental data. And so this is what we find about the self-renewal probabilities. And I'm going to talk quite a bit about these self-renewal probabilities. So in the long-term stem cell, that's given by 0.5. That has to be like this, because if it's not 0.5 at equilibrium, the population would be either expanding or declining. Okay, so that's what you expect from a population that can maintain the entire blood system. Now, the short-term stem cells, you can see, have a self-renewal probability that is less than 0.5 in these estimates. And the common interpretation would be that they cannot maintain themselves because the cells differentiate more than they divide. So if you leave them alone without the long-term stem cells, they'll differentiate out and disappear. And the next compartment down, the MPPs, multipotent progenitors, they have an even lower self-renewal probability, and that means they also cannot maintain themselves. But we dug into this a little bit more in detail. And this is shown in the graphs over here. So,
for understanding, you are still dealing with symmetry.
Probability of, yes, probabilities, yes. You could also put asymmetric division on top of that. It won't change too much. I think we've done that.
It will not have impact for computation.
Minor, probably. Yeah, we haven't put it in this particular thing. We've done in other studies. It didn't have much of an influence there. Yeah. Okay, so these are some simulations of this system that I showed you, and we are at equilibrium. Homeostasis, parameterized model has all the parameters of the experimental system. And what we do here now is we numerically or computationally deplete the long-term stem cell and see what happens in this model. And although you have these self-renewal probabilities, you can see that if you deplete the long-term stem cells, the system does not actually go extinct. The system re-equilibrates to a different equilibrium and is maintained. By what is it maintained? You can see here's the self-renewal probability of the short-term stem cells. So if you deplete the long-term stem cells, the renewal probability of the short-term stem cells goes up and dynamically adjusts to be 0.5. And this is the result of the feedback that is happening. So in contrast to what is the common interpretation, although at equilibrium you observe a self-renewal probability that is less than 0.5, if you deplete the long-term stem cells, it adjusts to be exactly 0.5. And now here, we not only deplete the long-term stem cells, but also the short-term stem cells. Now the MPPs kick in and they dynamically adjust to have their self-renewal probability go up to 0.5, and now they maintain the hematopoietic system. So this exercise tells you that although you have these self-renewal probabilities at equilibrium that you can observe and measure, the interpretation is not straightforward. Even if they're less than 0.5, it does not mean that they cannot maintain the hematopoietic system in principle, because if you remove the long-term stem cells, the rest of them adjusts as a result of feedback to maintain the hematopoietic system. Of course, eventually they will run out of replication capacity, especially if the MPPs maintain it, and because they don't have infinite replication capacity, but for quite a while they can maintain the hematopoietic system. So this is just schematically showing the same thing. These are the observed values at equilibrium here. Now you delete the long-term stem cells, and now this one adjusts to become 0.5, and this essentially becomes the stem cell then. Why is that happening? And this is explained here with the MPPs as an example. So MPP has, this is at equilibrium, the less than 0.5 self-renewal probability, and this is the result of feedback signals that are happening. And so I've split up this population of MPPs into two populations, one subpopulation, one that is like it's intrinsically replicating here in that compartment. That's the blue thing. They produce feedback factors upon themselves, but also you get influx from the long-term stem cells in here, and they produce, this influx produces additional feedback that reduces this value of P2. Now, if you cut that input by removing the long-term stem cells, you weaken the feedback signal, and as a result, the self-renewal probability rises to 0.5, and then they can maintain the system. And so these model predictions are consistent with data that are out there and can explain some of the data. So if you take transplanted short-term stem cells or MPPs in a mouse, you irradiate a mouse, get rid of the long-term stem cell, only put short-term stem cell in or MPPs, they can maintain a hematopoietic system for a while. Okay, so that was a puzzling observation, and this can sort of explain it. Also, you can take mice and you can get rid of the deplete the long-term stem cells or the stem cells in general, and also the progenitors. Over time, the long-term stem cells typically don't recover that much, but the MPPs, the progenitors do, and that can maintain a hematopoietic system. So this is consistent generally with observations that we see in mice. Okay, so this is sort of basic interpretation of data, of kinetic data at equilibrium, trying to understand how the system works at homeostasis. So the next step that we did is we want to incorporate evolution into the system and look at mutations. And we come back to this question is, what are the conditions for these chip mutants to evolve? In other words, a chip mutant is probably advantageous in evolutionary terms. What does it take for an advantageous mutant to evolve in this hierarchical system? So for that, we expand the model that we had so far to include mutants. So this is the original model that I showed you before. And now to put on top of that, another set of equations equivalent, but for mutants. So X is wild type, Y is mutant. And we assume that wild type and mutant differ in their self-renewal probability, because we know in these models that the self-renewal probability determines fitness. So a higher self-renewal probability gives a higher fitness to the cells. They will outcompete the other ones. How does competition work in here? Competition works in here because you share feedback signals. So both of these produce feedback signals and they can inhibit each other. And so competition happens through the shared feedback. So now the feedback goes from both of them, and the one with the bigger P essentially wins. So we're looking at advantageous mutants. The fitness is determined by this parameter S here, the increase in the self-renewal probability. Yeah.
What happens if we don't have any feedback? Can you compare?
Well, if you don't have any feedback, it's a very difficult model, because how do you get homeostasis? You have to have artificially have the P at 0.5, and they wouldn't be competing either. So they would be differentiation can happen. Yes, if you're self-renewal in the long-term sense is exactly 0.5, and you have constants. Yeah, you can simulate the same things, but you get different interpretation. And that's part of the interesting part of this model. So this, what I said before about the adjustment of the self-renewal probability, that would never happen without feedback. So that's a result of the feedback. So if you didn't have that, you wouldn't observe that. And in the evolution, it would be very difficult to model, because they wouldn't be a competition mechanism unless you put something else in, like a shared space or something for which they compete. That make sense? Okay. So, okay, advantageous mutants. We're interested in when can advantageous mutants rise. So we can place the mutants in different compartments here. We can place them into the long-term stem cells. So it's very unlikely for mutants to get generated in the long-term stem cell compartment, because we know they replicate very slowly, and so it's unlikely to generate them. We can place them in the short-term stem cell compartment. This is more likely. It's even more likely that they get placed in the multipotent progenitors, because they start dividing more and more if you go downstream. So we're asking, what is the fate of an advantageous mutant if you place them here, you place them here, and you place them here? And this is shown in these graphs over here. So there's a fate of a mutant when placed into the different compartments. We place the mutants into the long-term stem cells. It's an unlikely event, but if you do place them, we assume that they have a 2% advantage. This is actually a pretty big advantage, 2%. The mutants are in dashed line, the wild type and homeostasis are in solid line. So the mutants will start growing, but you can see that they grow really slowly. It takes 10 to the 4 days for them to kind of rise to appreciable levels. And the mouse only lives like for about 1,000 days, less than 1,000 days. So it's even if they get generated here, and even if they grow, they probably grow too slowly for them to be noticed. Now let's put them, the same mutant we put in the progenitors downstream, okay? Same mutant, same advantage, it's a 2% advantage. What we observe here is that the mutant fails. It goes extinct. It does not have a selective advantage in that compartment. So what does it take for the mutants to grow in that compartment? It takes a massive advantage. You can see here, we take a 25% advantage of a mutant, and now it grows, and it grows at a reasonable speed to make it to higher levels during the lifespan of the mouse. So what this tells us is that it's here they can get going, but it's a very unlikely event in the long-term stem cells, and they grow really, really slowly. In the downstream compartment, in the progenitors, it's much more likely to generate them, but they will fail if the advantage is not massive. So you need a massive fitness advantage for the mutants to start growing. So we call these things invasion barriers. And what we find is that there's a trade-off between likelihood of getting generated and the ability to grow the invasion barrier. So in the long-term stem cells, there is no invasion barrier. A mutant of any advantage will start to grow, no matter how tiny the advantage is. However, it's unlikely to generate a mutant here. And if they do grow, they grow really slowly, so you probably won't see them. On the other hand, if you place a mutant downstream in the MPPs, they're likely to be generated there, but there's a massive invasion barrier that has to be overcome. So you need kind of a monster mutation for this mutant to get going. And using the parameters that we have from the data, we can derive a condition. We can calculate what this mutant invasion barrier is. We can put a number for it. In the short-term stem cell, it's about 6%, and in the MPPs, it's about 20%. Both of them are huge fitness advantages. A biologically reasonable fitness advantage, like 1 or 2%, is already a large advantage. So we can say that there is this trade-off going on, and the more likely it is to place a mutant somewhere, the more difficult it is for that mutant to invade. So there's a certain resilience in the hematopoietic system against mutant emergence. So that's an important result, what we think. So why is that invasion barrier happening? And this is explained in this graph here. And the reason is that the fitness of the cells in the downstream compartment, let's say the progenitors here, is determined by two things. The first is the intrinsic replication of the mutant in the compartment. Okay, this is shown by these arrows here. So they all self-renew at a given rate, and the mutant has an advantage because it has higher self-renewal probability than the wild type. So that gives it an advantage. The second component of fitness is there's influx from the stem cells to the progenitors, right? They go from here, they differentiate out, and they go here. And the wild type cells are present in the long-term stem cell compartment. They will differentiate out, make their way to the progenitor cells. However, because the mutant is placed here, there's no mutant in this compartment. So it doesn't experience this influx. So this gives it an intrinsic disadvantage. And this gives rise to the mutant invasion barrier that has to be overcome. So that explains this barrier. Okay, so we've established using this model that there's an invasion barrier. It's difficult. The hematopoietic system has an intrinsic resilience to mutant invasion. It's very difficult. It's either very unlikely to produce the mutant and then invades very slowly if it happens in the long-term stem cell, or in the downstream compartment, it becomes more likely to produce the mutant, but there's this massive invasion barrier. So how can we get the mutants to take off? And we consider two possibilities here. One is aging, and the other one is what we call evolutionary niche construction. So aging, there's data out there that shows that aging results in a reduced influx from long-term stem cells into the downstream compartments. As a young mouse will have a strong influx, strong differentiation from stem cells to downstream compartments, an older mouse will have a weaker influx. Then there's evolutionary niche construction, where experiments have shown that mutant cells can induce inflammation, and inflammation increases the fitness of the mutants. So they increase their own fitness. So I'm going to explain that in a little bit more detail. So this is the aging business. As I said, you've got a young mouse. Thick arrows here show the strong differentiation from long-term stem cells into downstream compartments. As the mouse gets older, you can see that the arrows get thinner and thinner. It's because the rate of this influx becomes weaker and weaker. So that's one assumption that we're going to put in the model. The second assumption is the evolutionary niche construction. So the way it works for the mutant is you've got the stem cells, and eventually they give rise to the more differentiated cells. These more differentiated cells, they produce inflammatory mediators. That's a characteristic of these mutants that has been experimentally documented. And it has been further documented experimentally that these inflammatory mediators, they reduce the self-renewal potential of the wild type stem cells, but they don't affect the self-renewal probability of the mutant stem cells. So as the mutants become more abundant, they're going to be more and more of these inflammatory mediators, and that will favor more and more the mutant. So initially, when the mutant is at low frequencies, this won't make much of a difference, but if it increases, the fitness of the relative fitness of the mutants amplifies as a result of this. So we call that evolution in niche constructions because the mutants construct their own niche that increases, promotes their own evolution. I'm going to show that in a second, what happens in these models. So here we have the simulation that we had before, 2% advantage. We're going to consider the short-term stem cell as an example now, where we have the 6% invasion barrier, 6% advantage barrier. So this is what we had before. You take 2% advantage, a mutant with a 2% advantage into the short-term stem cells, they will go extinct. Okay, that's the invasion barrier. Now we put the effect of aging in, as I just described. Over time, we just assume that the influx of stem cells to the downstream compartment gets reduced by a constant amount at regular time intervals, which is shown here. Here it was constant, that didn't happen. So here now we have aging. And what we see is that the mutant now doesn't go extinct anymore. Because you have the reduced influx, this invasion barrier goes down and they can start growing. But you can still see that under the parameters given, they grow really, really slowly. Because by 1,000 days, which is the end of the life of a mouse, the mutant hasn't risen much. But if you now also put the niche construction on top of this, the mutants start growing at a decent rate, and they will take over within the lifespan of the mouse. And the reason is initially the growth is slow, but then you get the inflammation going. The inflammation increases the relative fitness of the mutants, and that gives a positive feedback, and that leads to the eventual rise of the mutant. So we think that this combination of aging and evolution in niche construction, both of which have been documented experimentally, can be a factor that helps the mutants to overcome the invasion barrier. So that was an interesting finding, and that's where we're at the moment. So we're trying to explain how these mutants pop up to start with, what gives them a selective advantage. And I think the interesting finding that we saw was that there is this invasion barrier, there's this intrinsic resilience in the hematopoietic system against mutant invasion, and that special conditions need to be met in order for the mutants to come up. And once you understand these special conditions, you can then try to influence these microenvironmental conditions in order to turn the fitness down of the mutants and get rid of the CHIP mutant burden, for example, by addressing inflammation, reducing the factors that select for the mutant stem cells. So, okay, that's where we are with this. So then going to the last part of my talk, I want to talk about spatial dynamics. So here we talked about the hematopoietic system, and we use basically ODEs that assume that cells mix perfectly well. And people usually justify that with blood because blood is a mixed system, but it's also not quite true because all the action of the stem cells happens in the bone marrow. The bone marrow is kind of in more spatial system. And so it's not all that mixed. And just in general, if you look at stem cell dynamics, we want to move away a little bit from the hematopoietic system and talk about it in general. If you have these dynamics of self-renewal and differentiation that go on in the ODE, as I described before, what happens if you formulate the same thing in a spatial model? Okay, so this is what we're going to concentrate on. We're going to get rid of the complexity of these different subcompartments in the blood and just consider stem cells and differentiated cells. Same structure, stem cells self-renew, they divide with the given rate R, they self-renew during that division with the probability P, with the probability 1 minus P, they become differentiated cells, and we focus on the feedback. Let's say differentiated cells can produce feedback factors that reduce the self-renewal of the stem cells. And that, if you, as I mentioned at the beginning of the talk, you formulate that in ODEs, that leads to a stable equilibrium, that leads to homeostasis. And that is the main feedback loop in this model that leads to homeostasis. You could have a different feedback loop from going on R that doesn't really contribute to homeostasis, but this does. So let's focus on this feedback loop. So in the ODE, you have homeostasis as a result of this feedback loop. What happens in space? So we look at a agent-based kind of model. And this is a sort of hybrid model. We've got kind of two levels here. You've got a grid, a true agent-based model where you place the cells. You've got stem cells and differentiated cells. Each spot in the grid can contain either a stem cell or a differentiated cell or be empty. And then there are rules to update this agent-based model. Rules are exactly the same as the assumptions in the ODEs. Okay, the cell can divide with a given probability. Upon division, it can be a self-reunion division with a probability p, or a differentiating division with a probability 1 minus p. Now, a differentiated cell can give rise to feedback factors. And these feedback factors are produced in a sort of parallel grid. And the feedback dynamics are then given by a patch model, where in each patch they're described by ODEs, and then they spread out and diffuse out to neighboring patches. And the feedback factors determines the fate of the cell in its particular location. So if you're a stem cell here, you look at the corresponding grid here, you see how many feedback factors there are, and this then determines the effective self-renewal probability of the cell. So the assumption is exactly the same as in the ODE. It's just in a spatial system. It's a very simple model, but now formulated spatially and not as an ODE. So what can we say about homeostasis here? So you've got the feedback on the self-renewal probability, and we look at what we say about homeostasis. How do we check what happens? Is we can increase the size of the grid, right? You have the size of the grid, that's the maximum population size that can be achieved in this spatial model. And so we can increase the size of this grid. So we start, this K is the carrying capacity. Let's say this is 10,000 here. This is the number of stem cells that you get. This is the number of differentiated cells you get. Now you increase that carrying capacity, you expand the grid, make it bigger, you get a larger number of cells. So it doesn't really lead to homeostasis anymore in the spatial model. And that's shown here. This is how big the grid is. This is the number of cells, differentiated cells, stem cells, and they go up as the size of the system increases. And that basically means that what limits the number of cells in the spatial system is really the grid size and not the feedback factor. So somehow things work different in a spatial model. The feedback doesn't really regulate the number of cells here. But there are other interesting things that are happening in this system. So when you do have strong negative feedback, you get interesting patterns emerging where you have clumps of cells separated by empty space. And these clumps of cells over here, these things, they consist of stem cells, which are in light blue, and differentiated cells, which are in yellow. And this kind of pattern almost reminds me of things like stem cell niches, where you have these little microcompartments where you have stem cells that differentiate out and the dynamics going on locally. So one thought is that maybe the feedback here leads to pattern formation that could give rise to these specific niches, although that's a speculation rather than anything else. And I would like to point out that the numbers look reasonable here. You've got stem cells that are in the minority, and you've got differentiated cells that are in the majority. This is how a healthy state looks like. Now we can reduce the amount of negative feedback inhibition in the system, and we find something interesting. Now the distribution of the cells becomes uniform. There are no clumps anymore. And now the stem cells are in the majority and the differentiated cells are in the minority, which represents a pathological state, because that's not how it should be. So it appears that while this negative feedback in the spatial model does not maintain homeostasis as such, it does influence the properties of the system. Strong feedback leads to spatial structures that lead to a biologically reasonable proportion of stem cells and differentiated cells. And if you have weak negative feedback, then this breaks down, and you've got stem cell enrichment, which corresponds to a pathological state. And this is an interesting observation. This is even more interesting here. You can plot a measure of this clumpiness against the strengths of negative feedback inhibition. So we take an approach that they use in ecology to quantify clumpiness of the system, where you divide the grid into little subgrids, and you look at the number of cells in the subgrids, and you look at the variance versus the mean. And if that is less than 1, that indicates a uniform distribution. If it's bigger than 1, that indicates a clump distribution. And you can see that as you increase the strength of the negative feedback, for weak negative feedback, you have a uniform distribution, as I already mentioned. But then there seems to be a threshold effect. When you cross a certain threshold in the strength of the negative feedback inhibition, it jumps to become clumped and to obtain these properties of the system. And so we don't really understand at this stage what causes this threshold behavior, but it is there and something to be investigated as we go on. So here's a little bit more detail on this, looking at how the system depends on some of the parameters. And the parameters we varied is the rate of feedback inhibition. And then, for example, here, the rate of the diffusion of the feedback factor across space. And each dot in this graph is the outcome of an individual simulation. And if there are the 3 outcomes that can occur, you can either have the uniform distribution, you can have the clump distribution, or the system can go extinct. And you can see that it sort of depends on these parameters somewhat. You've got, this is the interesting with the spatial clumps here. You can see that if the rate of feedback diffusion is very limited, it's very, very spatial, this becomes a very narrow parameter region. So you get either this uniform, this pathological outcome, or you get extinction and nothing much in between. But if the diffusion is a bit faster, then you get a wider area of this parameter region where this clump distribution occurs. That again indicates that the more spatial the system it is, the less this feedback kind of does something. And this tells the same story. This is the rate of feedback inhibition again against the decay of the feedback factors. If they decay pretty slowly, they're more likely to diffuse out. And this is identical to this outcome. You get a broader area of this parameter regime here. So this is just for more details of this behavior. So we've established that in this particular system, this negative feedback loop that results in homeostasis in the ODE does not result in homeostasis in the spatial model. So how can we get true homeostasis in the spatial model? And the obvious answer is you have to add more complexity to the model. And this is, we can just try out various things. One thing we did was we looked at a particular regulation mechanism that is given by this paper that I'm quoting here. That's in the epithelium, airway epithelium in mice. There are stem cells, which are the basal cells. Then the transit amplifying cells are secretory cells. And then you got terminally differentiated ciliated cells. And it appears that in this system, there's another feedback loop that is happening. And it's actually called a feed-forward loop. And the stem cells produce factors that allow their descendants to maintain their transit amplifying status. If they don't receive that signal, they differentiate out. Okay, and that is an intrinsically spatial mechanism because it depends on the vicinity of the transit amplifying cells to the stem cells. So the stem cells, they produce this factor. There's lots of it here. So all these cells that are around it will receive the signal, will maintain transit amplifying cell status or progenitor status. And as you move further away, the signal gets weaker and they start differentiating out. If you now plug that into the model, it is interesting because you do get true homeostasis. So again, you have this exercise where you vary the system size, vary the grid size, and you plot the number of stem cells and differentiated cells, and they're independent of system size now. There you get a blob of cells like this, and it doesn't expand. It kind of moves around a little bit and stays there. And so you've got true homeostasis. No matter how big the grid is, the population is going to be the same. So the message is that in the spatial model, you need more complex feedback mechanisms compared to the ODE in order to maintain homeostasis. This is an example that can achieve it. There are probably other examples that can achieve the same thing. The big message here is that you just need more. You need more complicated mechanisms. And now the next step that is ongoing work now is this is a very simple scenario where you have cells that kind of float around in the middle of nothing. You have stem cells and differentiated cells, maybe transit amplifying cells. They are by themselves there. But if you look at tissues or tumors, this is obviously not the case. They don't just cells don't just hang around in empty space and produce feedback factors that regulate their growth, but there are structural components there, which is called the extracellular matrix. And this can consist of fibroblasts, collagens called stroma. And this is a picture from a collaborator, Keith Chan, that images these things and shows you how these things can look like. And that's in the context of bladder cancer. These are bladder cancer grown on the back of a mouse, which are called xenografts. And the green here shows you the ECM, the extracellular matrix. And then there are stem cells that are in blue and differentiated cells in red, I believe. Yes, that's the right thing. And you can see that there are structures where you have these ECM components that kind of enclose the stem cells and differentiate cells. So you get pockets. So the population of cells is kind of subdivided by the ECM into little pockets that are surrounded by ECM. And the question is, why does that happen? And we think it has to do with particular feedback regulation mechanisms, the crosstalk between these different cell populations. And so we started to explore that with the similar agent-based model. The colors are different as before. The ECM is in yellow, and you get stem cells, progenitor cells, and differentiated cells. And you can see, and these are different simulations under different assumptions about feedback. It contains some complex interactions between the ECM and the tumor cells. There are feedback loops going on. And you can see in some cases, you get results that do not look like the experimental results. Here, the ECM kind of surrounds a blob of growing cells. This is not what we see, but other parameter combinations give you this kind of compartmental structure, these pocket structures that we see. And we're now in the process of working out those details to see under what conditions you get the experimentally observed patterns. And that brings me to the end of the talk. I just want to acknowledge people in this group, current and former group members, undergraduate, graduate, and postdoc. Even a high school student has been instrumental in this work, and then collaborators from math and from hematology who have contributed to this work. Thank you very much.
Thank you, Dominik.
Yeah. I guess I have one question about the R2 parameters in the OD model. It seems to me that from